The floor is yours.
Okay, thank you very much. My name is Leah Martin, and I'm doing my M2 internship on the effect of bacterial inoculum and alder leaf litter on the productivity and mineral use efficiencies of young alder and ryegrass plants growing in association and monoculture. And I am doing this internship between two labs, between the UMR Silva lab and the LAU lab at NCEA. And I'm working with Andy, a PhD student, and Severine and Pierrick, my supervisor. And I'm going to talk about my project today. So a little bit of background on the importance of the research that we're doing. So basically, as we probably all know, there's been increasing pressure on agriculture and forestry sectors to meet growing food and wood biomass demands. And then we also have the effects of climate change, which make things difficult. And that has led to increasing use of chemical or inorganic fertilizers. And in the most recent FAO report, they show a 44% increase in the global use of inorganic fertilizers between the year 2000 and 2021. And this increase has happened in all regions of the world. And while chemical fertilizers can be very effective, they can also create some issues of environmental problems, health problems, and damage soil structure in the long term. They're also not an indefinite sustainable source for plant nutrition, and it creates a lot of extra time and money for farmers and foresters to use these fertilizers. So the objective of my research is to test two alternative fertilization methods, looking at a biofertilizer treatment and alder leaf litter on young alder and ryegrass plants growing under agroforestry and monoculture conditions for the potential of these fertilizers to increase the soil nutrient availability and to be able to reduce the use of inorganic fertilizers. So in this project, we're looking at agroforestry systems, biofertilizer treatments, and leaf litter amendments, as through the literature we can see that they're promising alternatives for improving the nutrient cycling. So agroforestry systems can work and maximize nutrient cycling through niche partitioning and facilitation, whereas the different kinds of trees and crops you're growing can access different temporal or spatial niches and can help improve the nutrient cycling of the other plants in the system. The Pseudomonas biofertilizer we will be using, or we used, has been shown to increase nitrogen and sulfur in soil, and also the root surface area and length, which is important for nutrient absorption. And the alder leaf litter has been shown to improve soil nitrogen and phosphorus. So for the experiments, we conducted a short-term, 2-month-long semi-controlled pot experiment in the phytotron. We had 5 fertilization treatments, our bacterial inoculant of Pseudomonas, alder leaf litter added at 1 gram per pot, a combination of the bacteria and leaf litter treatments together, our traditional mineral fertilizer, and then a control without any type of fertilizer. And we have 3 modalities: the agroforestry modality with 1 alder tree and 4 ryegrass plants grown together, and then alder monoculture and ryegrass monoculture, and 6 replicates per pot. So we had 90 pots in total, and then another 4 control pots with just soil. So these are some pictures over the course of the experiment. So on the left, we have the T0, and at this time, the alder trees had already been planted for about a month beforehand to make sure that they were established and growing properly. And we had also added the leaf litter at the time we planted the trees. And then at the T0, we added the ryegrass plants and the bacterial inoculant and mineral fertilizers. And then you can see in the middle, after 1 week, there was a good amount of growth, especially in the agroforestry pot that's shown. And by the end of the experiment on the right, we were running out of space in the phytotron, and our plants had grown quite a bit, so it was time to harvest them. So the measurements that we took, we are looking at plant growth factors, including root analysis through WinRhizo, leaf pigmentation, the water use efficiency of each pot through the transpiration, the leaf number and area of alder, the fresh weight of our biomass, the ryegrass height, also looking at nitrogen, phosphorus, and sulfur content in the plant material and the soil, and ion analysis. Also, we are looking at the soil enzymatic activities connected to the carbon, nitrogen, sulfur, and phosphorus. So I have listed the enzymes that are related to those. And then these are the equations that I will be using to calculate the nutrient use and uptake efficiency for nitrogen, sulfur, and phosphorus in each of our pots. So we'll look at the dry weight and the nutrients that are in the plants and that were available in the system. And so in around the beginning of June, we harvested all of our pots, and this took a week with 6 people. We had 2 bachelor students working, and Andy and Severine and Pierrick, we were all working to collect our samples and separating roots and taking measurements. So you can see some of the pictures of all of that. And yeah. And now I have some preliminary results. We're currently in the stage of trying to finish up the laboratory tests by the end of the month and collecting the data. So a lot of it still needs to be analyzed and finished being collected. But one of the bachelor students did some tests with chlorophyll content. And so this is her graph, and you can see from her results that the bacterial and litter treatments and fertilizer treatments had significantly higher chlorophyll content than the litter treatment and control. And you can also see from the pictures of the ryegrass, just visually, there's more above ground biomass in the bacterial treatments and the fertilized treatment than the leaf litter or the control. And then some graphs from Andy, like you can see for the above ground fresh biomass, again, on average, the bacterial and fertilized treatments are higher than litter and control. And also the land equivalent ratio of our agroforestry pots is a little bit higher than the monoculture. So our preliminary results have indicated that the bacterial inoculant has positive effects on the plant growth factors, especially with the nitrogen, while the leaf litter treatment doesn't seem to have a significant effect compared to the control at the moment. But for our next steps, we have more analysis to do to confirm that. So we need to characterize the enzymatic activity in the soil, finish scanning the roots in WinRhizo, which hopefully will be finished this week. And then we can finish taking all the dry weights, which will be used for the nutrient use and uptake equations. And finish ICP testing, which will give us the results for phosphorus and sulfur content in soil, leaves, and roots. And then finally perform the statistical analysis to compare the different fertilization treatments and the agroforestry and versus the monoculture treatments. These are some of my references, and yeah, if you have any questions, thank you for your attention.
Thank you. So do we have some questions? There are not a lot of people, in fact. Maybe I can try. Do you have an explanation of why there is an effect of the inoculant, I think?
Sorry, why there isn't effective?
If you go to your last slides.
Yeah. This one?
Yeah, because can you maybe explain again, please?
Yes, so this is just the average amount of fresh above ground biomass in weight in grams for by each treatment, by the bacterial different fertilization treatments. So we can see that the above ground biomass for the bacteria litter and the traditional inorganic fertilizer treatment, there was a higher average in weight of the above ground fresh biomass than the litter and control. And then also when looking at the land equivalent ratio, the agroforestry one seems to be a bit higher, but we do need to do some like statistical tests on it to see if there's a significance. But these are just like some preliminary graphs.
But how did you calculate land equivalent ratio? Because I think in general it's around 1.
Mm. Yeah, so we took the amount from the monoculture of the ryegrass and the alder, and then divided it by 2 because they were in 2 separate pots, and then compared that to the agroforestry. But maybe the scale on the side needs to be put into a different scale. I'm not sure.
Because yeah, I think in general for the land equivalent ratio you have one value for the agroforestry treatment, and you will test if this value is superior or not to 1.
Mm.
In fact, in the calculation, you already take into account the pure uh, agronomic treatment and the pure algae treatment.
Yeah.
Because
I might need to relook at it. I'm not sure. We did it very quickly.
yes, okay. Because very basically, you just have to divide um, agroforestry, um, the yield of the grass in agroforestry divided by the yield of the grass in the pure treatment.
Mm.
It's the same for the tree. The yield of the tree in the agroforestry treatment divided by the yield of the tree in the monoculture.
Mm.
And in fact, you
Okay.
take
I think
into
we did
account,
both of them at the same time.
and you will have only one value for agroforestry, in fact, at the end.
Okay.
And this value should be close to 1 and hopefully superior, or significantly superior, or not to 1.
Okay. I'll relook at that for sure.
Okay. And I just want to be the only one to ask questions. So if somebody else wants to ask something to Leah, please.
Yes, I have one question.
Mhm.
Why did you choose Pseudomonas?
Yeah, so that is a bacterial inoculant that was patented by the LA lab, some researchers in the LA lab, and it seemed to have really promising results for improving the nitrogen and the sulfur. So we wanted to test that and in a controlled study and see. Maybe there were also some other reasons, but these are the main reasons to my understanding.
And is it possible to test other bacteria and to compare or not?
I mean, I'm sure it's possible, but I'm not sure like within this study. I think that there are plans to do PCR and to characterize the different microbial communities in the soil, but it's a bit beyond the scope of my master's project because my project is more focused on the nutrient use and uptake efficiencies. But maybe it will be a part of the bigger project since we're collecting a lot of data that won't necessarily be used in my master's thesis, but will be used for the larger project. So yeah, this could be possible. But I don't really know a lot about that aspect yet. So, and I don't know if there will be time for me to learn those things or not. I'm not sure.
Okay. And how many bacteria did you put?
That's a good question. I don't remember off the top of my head. I should have it, but yeah, I do have it written somewhere, but I don't know
No,
off
it's
the
not,
top of my head.
but it's because I
Yeah.
was wondering if it was a lot or not, and compared to because you were in a controlled condition and in natural environment, maybe the concentration could change due to the other bacteria already existing in the soil, and maybe there will some competition or stuff like that.
Yeah, maybe. I'm not sure. I mean, we collected the soil from the La Bouzou site. So that's where the soil came from. But then when within the pots, the system was like a kind of a closed system, and we tried to really prevent leaching. So it was yeah pretty limited to the area of the pot. So but yeah, I'm not sure, maybe there could be some interaction between other soil flora, fauna. I don't know.
Okay,
But
thank
it could
you.
be interesting.
But it's a nice work and nice presentation, very clear.
Thank you.
If I may, I saw on your picture some nodules on the other trees.
Yes.
Did you find a lot of this? It was the nodulation was efficient in the
Yes.
pot?
We did find some. I'm trying to find this slide now. Where was it? There we go. Yeah, we did find some nodules. It seems like we didn't calculate them yet. But it seemed like there were more present in the control and litter pots, and maybe a little bit less in the pots that seem to do better with the fertilization, the bacterial treatment, and the mineral fertilizer.
So you evaluated the biomass of the nodules?
Not yet. I mean, they are being included with the root system and the weight of that, but I don't think we're calculating them separately at the moment. But we might try to see if we can count how many per treatment. But yeah, at the moment they're in with the root system. So yeah, it was interesting because it seemed like maybe there were more root nodules in the control and the litter treatments that were not doing maybe as well with the biomass production according to what we've analyzed so far. But yeah.
And the nodulation was natural because when you bought the alder plants from the nursery, I guess maybe you don't
Yeah.
know.
Um,
They were already nodulated, or you don't know.
There were some very, very small nodules that we saw. But when we were planting some of the trees, there were a couple that had very small ones. But then when we harvested, there were definitely a lot more and they were bigger. But yeah, so so they should have had the ability technically to create the nodules.
Okay. And another question on the picture of your slide before, I saw that at the end of the experiment, the canopy were very dense.
This one.
Not this one, the one
No.
of the
Oh yeah,
chamber.
the other one. Okay. I know which one now. Yes.
Yeah, thank you. The one on the
Mhm.
right side. Do you think there were competition between the trees at this of different pots, I mean, at this time?
Yeah, I think there's a possibility, but I hope that there wasn't a big effect of that because we did try to spread the pots out as much as possible to try to eliminate the competition. So maybe at the very end, there started to be a little bit, but hopefully it wasn't a long enough time period to really make an effect. So we tried to control for as many variables as we could, but of course we can't control for everything. So hopefully we were successful in spreading out the pots to avoid that competition, but yeah, it's possible maybe there was a little bit at the end.
Okay. And concerning the below ground biomass, it's too early to have some data about it. Did you expect some differences between the different treatments
For
for the
the
below ground, the root biomass?
root biomass, yeah, I haven't really looked at that yet.
But you have the data.
We have the data for the weight of the root biomass is what you're asking about.
Yeah.
Yes. Yeah, we have the data because we are separate. We separated the roots from the stem and the leaves. So we have the weights of each of those, fresh and dry. But the thing is now we can't get the fresh and the dry weight for all of the roots until we finish scanning. And we still have like maybe 10 more or so left to scan. And so hopefully by the end of the week we'll be able to have that data. But we have to scan before we can dry them and then take the weights. So we're getting there, but not quite yet.
Okay. Thank you. So if somebody else wants to ask some questions, but maybe Pierre, Séverine, etc., you already know all of that. Okay. Thank you very much, Leah.
Thank you.
And good luck for the last analysis.
Thanks. Yeah.
If you need some help, don't hesitate to ask me. I'm very interested by the experiments, of course.
Okay. Yeah, we have a pretty good team. It's nice to not be working alone and have others, but I will definitely keep that in mind if things get too crazy. So thank you.
I can imagine.
Yeah.
Eh ben, merci à tous. On se retrouve la semaine prochaine pour un autre séminaire flash, le dernier de la saison, je pense. Bon après-midi.
Ok,
Thank you.
salut.
Merci Léa.
Merci Léa. Ciao, ciao.
Bye.
Merci. Bonne journée.